do hear the noon whistle.
Okay.
And so this is my opportunity to say that this is Steve Larson up here in Fort Atkinson, Wisconsin, with the Hoard Dairyman staff, bringing you our monthly webinar. We're very pleased today to have Chad Dechow, from Penn State. I'm going to get that name right one of these days, with us to talk about the new world of genetics, and it is an exciting new world out there, and we want to express our appreciation to the Neogen Corporation for their support of the webinar today. With those comments, I'll introduce Mike Hutchins, our colleague down at the University of Illinois, and also welcome again Jim Baltz, Mike's colleague down there as well, who helps us with the technical aspects of these webinars. So, Mike, I think we're good to go. Take it over.
Yeah, it's my pleasure and privilege to introduce Chad. Chad is an associate professor of dairy cattle genetics at Penn State University. Dr. Dechow got his bachelor's degree at Cornell University, his master's at Penn State University, and then his PhD at the University of Tennessee. He joined the staff there in 2003 at Pennsylvania. Prior to that, he had a small teaching assignment at one of the SUNY colleges there in New York State. His major area is improvement of dairy cattle health and well-being as he continues to look at breeding for higher levels of milk production and milk efficiency. So, Chad, I'll turn the program over to you, and it's all yours.
Okay, well, thank you, Dr. Hutchins and Steve. I really appreciate the opportunity for Hoards to come out and talk a little bit about the new world of genetics today. And when I was kind of thinking about this topic, there's a lot of different ways that I could go. And what I'm going to do is talk a little bit about crossbreeding and then spend the bulk of my time on genomic testing. And when I talk about genomic testing, I'm not necessarily going to spend a lot of time talking about what exactly we're testing in terms of how many SNPs there are on the chip and how exactly we generate bull PTAs from those, because that's been talked about a little bit in lots of different venues over the last couple of years. So I actually kind of want to spend a little bit more time thinking about kind of the new world of genetics, going beyond SNP tests a little bit, and also to talk about a little bit different way of looking at genetic progress than perhaps we have in the past. Not necessarily a new way, but looking at some formulas that maybe have changed how I've thought about it a little bit. But when we think about the new world of genetics, looking forward as well as looking back a little bit, certainly we have, it's not that we didn't do crossbreeding in the past, but the way that we've approached crossbreeding is a little bit different now in terms of more of a systematic approach in trying to develop a crossbreeding system. And so I do want to spend just a little bit of time in that area, especially when we look at some other industries like the poultry industry and the hog industry, that they really take advantage of crossbreeding. So I think it's something we could see more of in the future as well. So I did want to spend a little bit of time talking about that as well. But my first question, I wanted to start here to just get a little bit of sense with the audience whether they are involved with crossbreeding or not, and if they are they concentrating on domestic breeds like Jersey or Brown Swiss, foreign breeds, a mix of foreign and domestic, so for example Holstein, Jersey, Swedish Red Crosses or something like that, or D, I'm not crossbreeding. So I just trying to get a sense of the audience here.
Well, very good. We're off and running, Steve. I think I know where I'm voting. Have you got your vote
Well, I've got something in mind. Now, of course, for those of you that are advisors and consultants out there, you can respond on the basis of your clientele, what they're doing out there. But those of you who are dairymen or breeders certainly can talk about your own situations. So.
Well, I tell you, those Democrats, Republicans, Steve, are really working. We're up to 2/3 of the vote already, I'll tell you.
That's great response. And interesting response. So let's see what Chad has to say about those results.
Jim, let's close the polls and share that. And Chad, any comments, any surprises there for you?
Well, I guess probably a few. I was expecting maybe a little bit more in the I'm not crossbreeding column. We've got 56%, so a little over half not really involved in crossbreeding at all. And then 22% in the domestic breeds. And then basically between foreign breeds and foreign and domestic, if you add those together, that also works out to 22%. So, so going from there, that's actually a good place where I'm going next. I wanted to look a little bit at, it's kind of hard to know for sure how much crossbreeding is actually happening, because not all of the cows in the United States are on an official test program. So a lot of the cows we just don't know much about. And then on top of that, even when they are on DHI test, sometimes it's the way that sire information is entered and so forth, we don't capture a lot of the crossbred data. So maybe a different way to look at it is to look at what kind of semen is being used a little bit. And this isn't necessarily a perfect indicator, but I think it does tell us a little bit about what's going on in terms of commercial production, and especially for the foreign breeds, because we don't have any Montbéliards or Norwegian red cows in the United States. So if we're crossbreeding with those breeds, that means we're importing the semen. And so I think it's pretty interesting. You can see really in 2004 we weren't importing a lot of semen from these three particular breeds, a little bit of Swedish red. But then over the next couple of years, we saw a big spike in Swedish red, and peaked in 2006. And then a couple of years later, we saw a pretty big spike in Montbéliard semen that was imported. And then both of those have leveled out since that time, and we're now importing about 30,000 units of semen from those. And then of late, we've seen a pretty big spike in Norwegian red semen being imported, and it will be interesting to me to see whether that continues to climb, or if it follows the pattern of our other two breeds, where we get a spike and then a leveling off at a little bit lower level. Now, these numbers aren't perfect, because there was a virus in Europe that limited the import of semen in 2012, 2011, 2012. So that, to some extent, probably biases those numbers a little bit. But I think what it does tell you is that there has been, there was an initial spike, and then people that were using those breeds, the ones that liked them, continued, and those that, so we're importing 25 to 30,000 units of those, and that now there's a growing interest in Norwegian red, and we'll see where they level out. In terms of the, so that's a little bit on the amount of semen that's being used. This next slide gives a little bit of an indication of perhaps what we should expect in terms of daughter performance when we cross with these particular breeds. And this is, some of these bulls that we've imported actually have enough daughters where they have an official U.S. genetic evaluation. Not a lot of them, but a few. And if we go and we look at their PTAs, it can kind of give us an idea of what to expect when crossing with these particular breeds. So what we did, and this was from a couple of years ago now, but the results would be very much the same if we went and did it now, is we looked at how those bulls compare on a Holstein scale, and adding hybrid vigor into the equation as well. So as you can see, all three of these particular breeds, the cows would not produce as much as a typical Holstein, which I don't think is a real surprise, but very competitive for fat and protein, and also quite good for somatic cell score. So that's all very good. Now keep in mind, the Holstein bulls that we're using currently would be very high in all four of these traits as well. Holstein, the very best Holstein bulls are high for PTA milk, high for fat, high for protein, and also very favorable for somatic cell score. Where the top Holstein bulls can't keep up is in the fertility column. So these fertility rates are pretty much off the charts in terms of what would be compared to the very best Holstein bulls. So what we have here are these foreign breeds are, you know, maybe not elite for fat and protein, but reasonably competitive with a big boost in fertility levels. And there are certainly a lot of ongoing research trials, particularly one in Minnesota that is looking more closely at Montbéliard and Swedish Red. So over time we'll have an even better way to compare those crosses than we do currently. But I think based on the research results I've seen, this fairly well represents what we should expect, and in particular a big jump in fertility when we start to crossbreed. But we shouldn't, I think those breeds get a lot of attention because they're a little bit unique and different, but crossing with some of the more common U.S. domestic color breeds is also can work in a lot of different situations as well. And so perhaps the study that's kind of looked at both Brown Swiss and Jersey at the same time was work by USDA, excuse me, and this was presented in 2006. And what I've placed here are their differences in net merit and cheese merit on a Holstein base. So basically this 0 line that you see here would be the net merit or the cheese merit of the average Holstein cow born in 2005. And then anything above the line means that the crossbreds were above average, and below the line means that they were a little bit below average. So as you can see on a net merit basis, the first generation Jersey and Brown Swiss Holstein crosses were very competitive with purebred Holsteins. Of course, the question is, what do you do after the first generation? And for a while, the most common question I would get from dairy producers would be, I bred my Holsteins to Jersey, now what should I do? And hopefully we kind of think that through before we actually go down that road, but in the net merit scheme, there's a couple different options. One, we could add a third breed to the mix and do a 3-breed rotation, and when milk's a little bit more valuable, at least pounds of fluid milk, then those Holsteins still outperform the 3-breed rotation. But after we got out into the cross a little bit, Holstein times Brown Swiss times Jersey, those cows performed very well. So based on this type of data, we generally recommend a 3-breed rotation. And over here in the cheese merit side of things, where protein in particular and also fat are more valuable, you can see those 3-breed crosses tend to do fairly well compared to breeding your, for example, this purple column here would be breeding your Holsteins to Jersey and then breeding that first generation cross back to Holstein. So when protein's a little bit more valuable, those crosses produce more fat and protein relative to milk yield, and they do fairly well. So based on that, my summary of crossbreeding is that it gives us one more option to match the type of cow that we have with our management system and milk market. And if I think we've gone incorrectly in the past, it's that we've tried to select one cow that suits everybody's purposes and tried to maximize milk yield and type for our Holstein cows, and that works well for some management systems and not for others. So for example, if we've got an older freestall barn with smaller stalls, we might be able to use crossbreeding with Jersey and in one of the red breeds to create a cow that's a little bit smaller and that fits those types of stalls, and it's a little bit more comfortable for the cows. On the other hand, if we have very large stalls, then maybe we want to think about using some larger breeds, and the Brown Swiss have fit in there fairly well in terms of their competitiveness with pure Holstein cows. If there's one thing that you're going to improve with crossbreeding, it's fertility. And we generally now recommend 3-breed rotations. And we use 3-breed rotations because it kind of helps us to keep high levels of hybrid vigor for fertility and health, but at the same time keep enough of that Holstein influence in there to keep production levels fairly high. With that said, I think that there are a few different breeds that are economically competitive. We've got Jersey and Brown Swiss in the U.S., and then the 3 foreign breeds that we've talked about. We haven't seen a lot of head-to-head comparisons for these types of breeds. We've seen different crosses compared to Holstein in research trials and in various research herds. But we haven't, for example, seen Holstein-Montbéliard crosses compared directly in the same herds to Holstein-Brown Swiss crosses on a large-scale basis. So it gets a little bit tricky to know exactly what to recommend to folks because we haven't seen those direct combinations. And we probably won't see a lot of that. It'll take a long time to fill in the pieces to the puzzle because there's just not quite enough research in that area to really be able to have confidence in what to expect when we compare specific breed combinations. But with that said, I think that the crossbreeding results from the last few years have definitely demonstrated that it can work in a lot of different types of situations and really help match our cows to our facilities and our milk market fairly efficiently. So now we're going to, so that's one area where I think genetics is kind of changing a little bit. The other area is obviously with genomics. And so we're going to shift gears and talk about genomic testing a little bit. And I apologize in advance if what you had hoped to learn was basically kind of the different types of SNPs and how to test your cows and those types of things. I guess I was thinking a little bit beyond that a little bit and thought that a lot of folks have already been exposed to that. So I wanted to give my thoughts on genomics down the road in the future a little bit more completely and maybe help folks understand what happens to Bulls proofs as we go from genomic proofs to daughter-proven bulls and understand why we see some of the changes that we do in those Bulls proofs. But in terms of the number of animals that have been tested, you can see over here in the left-hand panel, we're starting to see really a pretty large increase in the number of females that have been genotyped, particularly for Holstein and Jersey. I think the high milk price helps that a little bit. We've got a little bit more money that we can afford to genomically test our females a little bit. Brown Swiss, still less than 1,000 cows per year, so they haven't quite kept pace on the genotyping of the female side. And then over in our right-hand panel shows the number of males that have been genotyped based on their when they entered AI, and you can see we really 2009 was when we officially released the first genomic evaluation. However, we also the SNP panels that were used to do the genomic testing were available in 2008. And we tested quite a few animals initially to try to build up our database in order to do genomic testing. And so you can see we've seen a pretty large increase in the number of males, and in particularly starting in 2013, dairy producers could test their males. Prior to that, the testing was limited to some selected bull studs. But that licensing agreement relaxed in 2013, and we're starting to see a little bit of a jump as producers genotype their own males. One thing that I think is interesting, though, is this purple line here, and this is the number of bulls that are entering active AI service. And you can see that's remains fairly level, about 2,000 bulls. And initially, I think folks thought, well, we might see a drop in the number of bulls just because, well, we're only going to bring in bulls with high genomic test results, and we may see a reduction in the number of bulls that we progeny test. But what we've seen is that we're starting to use those bulls. Producers are interested in using lots of young bulls early on, so that's kind of counterbalanced it. So we're seeing more genomic testing, but the number of bulls entering AI is not necessarily changed too dramatically. But where genomics has been a huge advantage is shown in this slide. I'll concentrate over here on the left-hand panel. This is the generation interval, or how old were the sire and dam when how old was a bull's sire and dam when he was born. And previously, the sire was almost 7 years old when his offspring were born, on average, and the reason for that was simply that we needed to have an accurate proof for a bull before we decided who we were going to use for contract matings on the AI side and so forth. That has very quickly declined. It's now down to 3.5 years, and I expect that it'll continue to drop even further from there. The average age of our dams has also declined. It was 4.5 years in 2005, so this would have been, this period out here would be before we had genomics. And then in 2009, genomics were introduced, and then you can see 2011, 12, 13 is where it's really starting to take full effect. On the right-hand panel, we see very similar type of thing, and particularly on the sire side, the paternal grandsire up here in blue. And this red line is the paternal granddam. We're seeing those the ages of those when their descendants are born have declined quite a bit. So, and we'll look at a genetic response formula later on that'll help to clarify perhaps why that's so advantageous. So in terms of what we've seen so far, we haven't seen the number of AI bulls shift dramatically, but how they're used is quite a bit different, and we're using them at a lot younger age than we were in the past. We're also seeing a higher percentage of bulls that enter AI service are the result of embryo transfer and IVF. And so we're seeing an accelerated rate of genetic progress. It's a little bit too early to determine how much the rate of genetic progress has accelerated. There have been some early attempts to quantify it, but those projections are based on a bull's genomic proof, and we really haven't seen bulls with daughter proofs that enable us to determine how quickly we've accelerated genetic progress just yet. Okay, so I want to spend a couple minutes talking about answering this question: can we be confident in genomic young sire evaluations? And I'm going to start with this panel here, and this chart here was included in a Hoard's Dairyman article a couple months back, and what we did was we just took the top 150 genomic young sires, which are these blue dots, based on their April 2011 net merit, and we restricted those 150. They had to have at least 50 daughters in April of 2014. And we want to look now that they have daughters, how does their ranking in number compare to these daughter-proven bulls. So these were the top 150 daughter-proven bulls in April of 2011. So if you look here, basically to make the top 150 in 2011, a bull needed to have at least about $525 of lifetime net merit in order to qualify, whereas a daughter-proven bull was $325 lifetime net merit. And on average, based on their 2011 proof, we expected the genomic young sires to be about $150 superior for net merit than our daughter-proven bulls. So based on those numbers, obviously the better group of bulls was the genomic young sires. However, when we go over to 2014, the story changes just a little bit, and now you can see on this y-axis is their current ranking. And on average, the genomic young sires are still the better group of bulls, but instead of by $150, it's by $60. So the difference between our genomic young sires and our daughter-proven bulls has basically gotten a lot closer. And when you look at the very top of the list, you can see there's almost as many daughter-proven bulls up there as there are genomic young sires. So as a group, the genomic young sires are better, but the difference between what we expected to happen and what has actually turned out now that we have daughters has shrunk quite a bit. And I'm going to talk a little bit on the next slide about two aspects of this. One is accuracy. And when I say accuracy, I'm basically asking ourselves, how much confidence do we have when we compare one genomic young sire to another? So you can see here when we look at this, looking at just these blue dots in the scatter plot here, our best genomic young sire, now that he has daughters, turned out to be that he really was the best. Our second best really was the second best. This group here's held up fairly well. When we get down the list a little bit, we see a lot more re-ranking. But in general, our genomic proofs, even though those bulls did not have any daughters, did help us to identify the very elite young bulls. So that was, from my perspective, it's very successful. Now, the amount of re-ranking here also suggests to me that, for example, if you use this bull down here that's plus $650 in net merit, and he turns out to be only $250 in net merit, if you used a lot of that bull, you're not very confident in genomics any longer. So that does emphasize that we need to continue to spread our risk a little bit. But to me, that's reasonable accuracy. The second term that I'll talk a little bit about is bias. So, and that, you would think accuracy and bias mean kind of the same thing, but not quite. Accuracy is how closely ranked they were from our projection to where they ended up. Bias is more, did we overestimate or underestimate their proofs? So this line here, this black line, if all the bulls had lined up perfectly compared to what we expected, they would all fall on this black line. Anything above this line means the bull was better than we projected, so they were underestimated in 2011. And anything below the line means that their genomic proof was overestimated in 2011. So there was a over bias, if you will. So when you look at the daughter-proven dots, you can see for the most part we've got an equal number above and below the line, which is what we expect to happen. There's one real outlier at $425 in 2011 that turned out to only be plus $50, so he was the exception. But generally speaking, equal number of red dots above and below the line. Where I have some concern is when we look at the genomic young sires, and you can see a lot of dots below the line, so we had a pretty significant upward bias in those genomic young sire proofs. So in terms of the early results to this point, I think that the ranking among young sires is reasonably stable. It's not 100%, but the reliabilities are not 100% either, so we weren't expecting them to be perfect. So the accuracy part of it, for me, has held up fairly well. Perhaps with the exception of productive life, that trait's been a little bit of a challenge to get correct, and part of that is because market conditions change. If beef prices are high and milk prices are high and you've got lots of heifers, then productive life becomes a little less valuable and changes the type of cow we're culling and when and so forth. So that trait is a little bit of a challenge in general, and it has been a challenge for genomic evaluations. So even though I think that we are reasonably accurate, there's still a lot of bias, and I always get lots of emails to disagree with me when I state this, but I still, looking at that last chart, still stand by it, and that's that we should not be comparing daughter proofs to genomic young sire proofs just yet. Until we reach a point where we've stabilized and don't see that drop any longer, it's an apples to oranges comparison, and we should not be making that comparison just yet. Now, we can, as a group, say, well, we expect the top 100 genomic young sires to exceed the top 100 daughter-proven bulls, and as a group, that is a fair statement, but by how much we expect them to be better than the daughter-proven bulls, I don't think we can yet state with any amount of confidence. The second issue is that some young bulls, when they first get their daughter proof, keep in mind, particularly for productive life, we have to have daughters grow old and die before we really have a true daughter evaluation. So we've seen some young bulls that on their first proof looked really good, but then when their daughters started to get a little bit older, their productive life proof dropped and their genetic evaluation dropped as well. So it takes 2 or 3 genetic evaluation cycles to really have a good handle on a bull's, how his daughters are performing. So next, I want to move in, and this is perhaps a little bit moving beyond making direct recommendations and looking at why we see a little bit of this change happening. And we're going to start here with this gene called DGAT1. DGAT1 is a gene that's involved in fatty acid synthesis in the mammary gland and other tissues as well. And basically it has one small role. It is to take a diglyceride, so you can see in that little chart there, and we're not going to go into depth into that chart, but we've got that diglyceride with 2 fatty acid chains, and then that DGAT helps to change that into a triglyceride. So when you go to the doctors and he does a triglyceride test, that's what he's testing. How efficiently are you making triglycerides? And there are 2 versions of this in cattle. So there's a small snip of DNA sequence, and the DNA codon GAA codes for a lysine amino acid, and GGC gives us alanine. And lysine is associated with high fat production. Alanine is associated with low fat production. And just looking at this quickly in terms of these charts here are what USDA produce that indicate where the big effects that impact a trait are located. And this is based on a genomic test, and these 4 panels each have about 61,000 dots on there. And you can see that for Holstein and Jersey, we see a very large effect on chromosome 14 that impacts fat production. That is the DGAT effect. We do not see that for Ayrshires and Brown Swiss, and that's simply because both of those breeds, most cows have the low-fat version, and so there's not as much variation in the DGAT gene in those 2 populations. So comparing in New Zealand was where a lot of this initial research was done. This is the DGAT1 effect in Holsteins, Jerseys, and they did have a few Ayrshires that had 1 copy of the allele, so they included that as well. And you can see on the x-axis, or the number of high-fat alleles, as we go from 0 to 1 to 2, we get about 26 more pounds of fat from our Holsteins, about 22 pounds more fat from our Jerseys. On the other hand, as we go from 0, 1 to 2 alleles, we get less milk production. Negative 600 pounds for milk, negative 300 pounds for Jersey. And the one thing that I want to point out on this slide is that the DGAT1 gene has a similar effect in all the different breeds, but that the magnitude of that effect is different. 600 pounds versus 300 pounds in Jerseys. And so what that tells us is that DGAT1 gene, going back to that earlier slide, it really doesn't do anything by itself. It's part of a long process, and it's one role in that process. So no gene has an effect by itself. They all work together. And I also wanted to look real quickly at what happens with protein. And since protein yield is so tightly correlated with milk yield, as we go up towards 2 copies of the high-fat gene, we see a decline in the amount of protein that we're producing. But in Jerseys, you'll notice 1 copy was associated with actually a little bit of an increase in protein yield. And I've just put a black line in there that shows us where the 1 copy would be if it had only an additive genetic effect. So this difference that we see here, we call that the dominance deviation. And we don't really fully directly account for dominance deviations in our genomic evaluations. So we'll come back to that aspect later. But that's one reason our genomic predictions aren't perfect is because we don't account for gene interactions and dominance. The second issue is that we don't actually test for DGAT. DGAT1 is a gene that was patented. Now, because of a recent Supreme Court decision, that the effects of that patent will change over time, and we won't have to worry about that patent. But what it has meant for the past few years is that to genomically test for DGAT would increase the cost of genomic testing substantially. So what we did was we looked for DNA markers, and those DNA markers that we test are called SNPs, or single nucleotide polymorphisms, that flank the DGAT1 gene, but we actually didn't test DGAT1 itself directly. So that works pretty well, testing around the DGAT1 gene. But what happens if we happen to get a recombination where we have SNPs that are associated with the high-fat gene and SNPs that are associated with the low-fat gene? Because we're not directly testing for DGAT, we're not sure what to do, and we would basically call a cow that inherited this average when in fact she got the low-fat gene. So that's one of the challenges with the way that we do our genetic testing and not being able to really pinpoint the exact causative mutation. So I've gone through that just to give you a little bit of an idea why I think that overestimates of genomic proof should not surprise us. And one of the challenges is that no gene can function independently. It's an integrated system. Our whole genome is working together. So when we change one part of the genome, the effect of a particular gene will vary. So for example, DGAT1 has the same general effect in Holsteins and in Jerseys, but the magnitude of that effect is different because the rest of the genome has been altered. And I tend to think that a lot of it has to do with the amount of nutrients that are available. So for example, a Holstein can produce more pounds of milk because she can eat a little bit more than a Jersey, so the effect of DGAT1 is magnified in Holsteins compared to Jersey. And that's true of all genes, not just DGAT1. The second issue is that we don't test the causal mutation. In most cases, we don't test the causal mutation because we don't know for sure what it is, what exact gene is responsible for a difference in two particular traits. And in terms of DGAT, it was because of not because we didn't know where it was, but because of a patent issue. And then lastly, we're ignoring that dominance deviation. So when you put those things together, we should expect to overestimate our proofs just a little bit on average. So I'm going to shift gears here again, and we're going to look at, for commercial producers, who we think would benefit the most. Would they benefit most from testing genomic young sires? Using genomic young sires in their herd? Should they use daughter-proven sires, or see they're both good options?
Okay, well, very good, Steve. I think we're back to our voting again to see what's going to happen with our votes, and the lines are now open, so let's be voting here. Our time's rapidly going, Chad. We're down to about 15 minutes of total time left, so...
Yeah, I'm going to have to hurry through some other things. I apologize for that.
No problem. Just so you're aware of that.
Well, the people vote fast, so we'll be right back with you here very shortly.
Well, I'm a little surprised, Steve, in the vote. What about you?
Yeah, well, yes, I am a little bit. Yeah.
Maybe Chad's not surprised. Anyway, let's close it off, Jim, and we got 2/3 of the votes in.
And away we go.
And Chad, what's your take on this?
Well, 60% of folks agree with what I would argue, which is that they're both good options for commercial producers, and I'll go through why I think that is the case here going along, and it just depends on what the commercial producers are interested in. So most folks appear to agree with me, but I want to start here with a formula, and this formula is basically just telling us how much genetic progress do we make per year. There are 4 factors that determine how quickly we make genetic progress: reliability, selection intensity, which is just are we picking the very best top 10% of bulls, the top 1% of bulls, the amount of genetic variation in a population, which we really don't have any control over, and then generation interval. So obviously we saw the slide earlier that showed that generation interval is going down, which is good because that is in the denominator and we want a low number there. What geneticists have done is they then partition this formula out into 4 what we call pathways of selection. The sire of sire pathway, this would be what the bulls that the bull studs use to make the next generation of bulls that they're going to sell to producers. So these are the very best of the best bulls. And then the cows that they mate them to are the very best of the best cows. We have traditionally the commercial herd considered what they're doing on as the dam side here in the bulls that they buy from the bull studs and breed their commercial cows to. We've considered the sire of the dam, and then the dam of the dam would be the cows in their herd that they're breeding. So when you think about this generation interval, if we use a lower generation interval for our sires of dams in our commercial herd, traditionally we've thought that that would accelerate genetic progress. But there's that formula, the 4-path model, was developed in 1950. Okay, so this just shows what would happen if that formula is correct. You are using young sires over time. You've got a lower number in your denominator, so your rate of genetic gain is going to be much faster than what we see for proven sires. So because of that, over time, the difference between you, who are using a young sire, and your neighbor that's using a proven sire would grow over time, and there would be a very big disadvantage to using proven sires, and then everybody should only use young sires. The problem is with that formula, and we kind of forgot the details over time, but that formula was derived for a closed population. If you're using AI semen, you're not a closed population. If you're using your own herd bulls and not importing any semen, then that 4-path model is correct. But for most people, it's not. So I just want to show you just a small little example here to show you what would happen. If I here in this first column, I'm going to year 1 through 10, we're going to use sires that each year are $50 better than the year before. I'm aggressive and I'm going to use those bulls now that they're young sires. My neighbor is going to wait 4 years, but he's going to use the exact same bulls, he's just waiting 4 years. Okay, so you can see in year 1, I'm breeding to an average cow, so my heifers that result will be plus $50 in net merit. Year 2, 100, 3, 150. In year 4, I'm using bulls that are plus 200, but I'm breeding him back to my heifers that I generated in year 1. Okay, and they pass on half of their genetic merit, so that gets us to 225, and then so on. My neighbor, using the exact same bulls but waiting 4 years, gets the exact same rate of genetic progress, only it's delayed by 4 years. So really we should expect commercial herds using genomic young sires or commercial herds using proven bulls to have the exact same rate of genetic progress as what we see in our commercial population. Now there may be a difference in lag, so my neighbor is 4 years behind me. Now this is kind of the worst case scenario for my neighbor because I'm not accounting for the fact that he's waiting 4 years to use bulls that have higher reliability, and the bull studs, after they figure out which bulls had genomic proofs that weren't accurate, are eliminating those bulls, so he has effectively a higher selection intensity, which will close that gap even further. So I'm not going to go over this slide in the interest of time. I'm going to switch here to some results from a simulation that basically looked at a few different scenarios. In scenario 1, we were using proven bulls in the elite AI herds and proven bulls in the commercial herds, and you can see that's the black and the gray line. This was for protein yield. Okay, and then in the second and third scenarios, we're using genomic young sires at the bull stud level. And you can see that green line there at the top, that really accelerates the rate of genetic progress beyond what we had in the progeny test era. So obviously from that perspective, the bull studs need to be using, for the most part, genomic young sires in order to accelerate genetic progress. On the other hand, the herds using the commercial herds using daughter-proven or genomic young sires are both going up at the same rate as the nucleus herd. It's just that there's a difference in lag. When we look at daughter pregnancy rate, because we're making a slower rate of progress in the elite AI herd, it is actually possible for the commercial herd using daughter-proven bulls to be even ahead of what's going in the elite AI population, because daughter pregnancy rate evaluations have lower reliability, and if we wait for daughters, that increases our reliability. And the other issue is that we weren't making fast genetic progress for daughter pregnancy rate in the elite AI population. So in that particular case, for that low heritability trait, commercial herds were better off actually using DPR. And of course, we don't have separate AI populations and commercial populations. We overlap a little bit. And this slide is just showing that what we expect to see is that males, which are the elite population, should be a little bit ahead of females, and that's what happened in our simulation in the left-hand panel here. And then if we compare that to what's actually happened over time, you can see the sires are always a little bit ahead of the cows. So essentially the sires that the bull studs produce set the pace and then pull the commercial population along with them at the same pace. So there's always a little bit of a lag, but they're both increasing at the same rate of gain. Okay, so rate of progress is going to be identical whether we're using whether we're using genomic young sires for our commercial population or we're using daughter-proven bulls in our commercial population. There will be a little bit of a difference in genetic lag, perhaps. So for those that are using genomic young sires, they need to continue to worry about spreading their risk, which is a little bit easier for large herds than small herds. But I also want to point out there's more than one way to spread risk. So a lot of commercial herds, larger commercial herds, don't really want to worry about a large semen inventory, so they've been hesitant to spread their risk because they think that means it's going to be more difficult. They're going to have to worry about a lot of bulls and determine what cow am I going to mate to bull X, what cow am I going to mate to bull Y, and they would rather just breed all their cows on off-sync day to one particular bull. Well, that's fine. So what those herds should do in that case is to use one or two bulls for a short period of time and just keep turning their bulls over rather than using a little bit of many bulls over a longer period of time. So there's more than one way to spread risk, and if you're using genomic young sires in particular, that's still the message that our commercial producers need to hear, is that they need to spread their risk a little bit. So commercial herds that are not marketing, if they're watching their calving traits on their virgin heifers and they're spreading their risk and they're willing to use a bull some bulls that are going to be misses, but usually they aren't going to be terrible. Going back to that chart way back at the beginning, the scatter plot, even those blue dots, those genomic young sires that turned out to not be as high as we expected, they still weren't terrible bulls. So if you're spreading your risk, using genomic young sires can work well for you. But I'd also like to get the message out there that you do not have to use genomic young sires if you're more comfortable using daughter-proven bulls. You're not going to fall behind your neighbors by doing that. Okay, so I've used up most of my time, so I'm not going to spend a lot of time on these next few slides. I put these in here as kind of looking down the road at the genome. So I'm going to go through these fairly quickly and just highlight a couple of things here. And the first thing that I want to highlight is that the genome is really only a small part of that is actually part of a gene. So about 2% of our genome are actually genes that produce the proteins like DGAT1 that carry out various functions. The rest of the genome is actually regulates the 2%. So I think of it this way. Genes are our computer hardware, if you will. And so if you have a computer without any software on it, you can turn the computer on, but you can't do anything with it. The rest of the gene, the other 98%, is what makes our genome work. Okay, and we've underestimated that 98%. We used to think it was all junk, and that was actually used as an argument against intelligent design and creation and those types of things. And now we find out that that junk is actually not junk after all. We now have, in terms of new developments, we've identified 28 million variants so far. So rather than testing for 90,000 SNPs, in theory, we could test for many million of SNPs, and maybe over time that will become important. In the near term, it's not all that important because it just doesn't increase our reliability very much. We're actually capturing most of the genetic variation using, we can use 45,000 SNPs, 61,000, 74, or 91,000. And when researchers add more SNPs, it does not increase the reliability of the genomic evaluations. So we're not really able to take full advantage of adding more information to the system just yet. So 28 million, we know there are at least 28 million SNPs available, but most of those aren't going to help us in the near term. One thing that might be able to help is recent research has started to look at trying to account for that dominance deviation like we looked at with DGAT1 a little while ago. And that did explain an appreciable amount of the variance that we see in genomic testing. So as we're able to use that type of information a little bit more fully, that could improve the accuracy of our genomic evaluations over time. These next three slides are a little bit, they're way out there, but one thing I wanted to point out is we've tended to think of the DNA code as producing an amino acid sequence. That's true. It is used to produce proteins, but a lot of those amino acids, a lot of those codons that tell us how to build a protein also help to regulate the gene and determine when a gene gets turned on or turned off. Because, for instance, we don't want our genes that express heart tissue to be expressed in our brains and vice versa. So there's actually something called dual-use codons, and they actually produce proteins at the same time as regulating when those proteins are produced. So the DNA code is much more complex than we could have ever imagined. There's also differences in DNA methylation that determine how our genome is packaged. So you can have two individuals, like a clone or an identical twin, that have the exact same DNA sequence, but their genome is packaged differently, and it alters how their genes are expressed and where their genes are expressed. Some of those differences are heritable as well. So we've got a little ways to go to fully understand the genome. One last slide here is looking at the ends of our chromosomes. We have a protective cap that gets shorter as we get older, and we've done some early work suggesting that cows with really short, we call this telomeres, this protective cap at the end of our chromosomes, and cows with really short telomeres have a little bit heightened risk for being cold. So here's my slide with the take-home messages. We're seeing a little bit more in the new genetic era, a little bit more crossbreeding. Genomic selection has dramatically altered dairy cattle selection progress and made progress faster in our elite herds. Our commercial population is going to be pulled along at the same rate, so that faster genetic progress that we make by using genomic young sires in the AI population will help us to make faster progress in the commercial herds as well. And then one final thing I'd like to leave you with is that I think we're just beginning to unlock the genome's secrets. So I don't think we should have an expectation that a genomic test is going to tell us everything, and for several years yet. We've got a long ways to go to really fully take full advantage of what we're able to do through DNA testing. And with that, I'm happy to turn the program back over to Dr. Hutchins.
Yes, and Steve, we'll let you kind of wrap it up here.
Yes, okay, Chad. Well, thank you very much for a very interesting webinar. You've helped us put crossbreeding and genomics in perspective in terms of how they're changing and how they're affecting and will affect our breeding programs on farms. You've done a great job of covering a lot of ground, and we sure appreciate that very much, and we also appreciate the support of Neogen Corporation today as being our sponsors. For those of you with us today, we thank you for your participation. We should remind you, especially if you're new, that in a few days you'll be receiving a very short survey by email, which we appreciate your responding to help us keep our webinar series relevant to your interests and needs, and so we will appreciate your help on that. And of course, this webinar, after a few days, and all of our previous webinars are available, archived by going to hoardsdairyman.com and clicking on the webinar link, and about an average of 10 people every day, 7 days a week, look at one of our past webinars. So we get a lot of interest out of those, or in those, and we appreciate that. And of course, we always appreciate this, the help of Mike Hutchins down at the University of Illinois, our co-host, and his sidekick down there, Jim Baltz. Just looking ahead to the next month, September 8th will be our next webinar, Getting Them Bred, an update on reproductive technology and theories by Matt Lucy at the University of Missouri, and they'll be sponsored by the Bayer Corporation. And then in October, we'll be looking at the results of FDA's antibiotic residue milk survey that we've all been waiting for some time. With those comments, Mike, I will ask you to see if there are some questions or comments that Chad can respond to.
You bet, Steve. We do have a number of questions here, Chad, so get ready. Here comes the speed round. The first question's a bit long, and we're off and running, and you want to hit the next PowerPoint. We want to make sure our sponsor gets good airtime here, so Chad, if you want to click that in Neogen, thanks again very much. Without our sponsors, this wouldn't be happening. A comment here, a number of units of semen sold by NAAB do not include substantial units of semen sold from Montbéliard and Viking Red in the U.S. directly by Creative Genetics out of California. They're not members of NAAB, but include only units sold by select sires. Therefore, your units of semen on those breeds are somewhat incomplete. Do you want to make a comment on that, Chad?
I was not aware that those weren't included in the statistics, so I apologize for that, and so that those numbers would not be perfectly accurate then.
Okay, let's go to another one. Are there breed differences in the confidence using genomic young bulls? In other words, is there a difference between the breeds?
Yeah, well, obviously for Holsteins we just have a lot more data, so we have the most confidence when it comes to Holsteins, followed by Jerseys, which would have the second most amount of data, and then Brown Swiss, and then Ayrshires, probably just a little bit less than Brown Swiss.
Okay. Next one is most young sires were overestimated in 2011, you pointed out, when it was correct in 2012. In other words, can we expect to have that same effect in 2017?
That is, if we knew for sure what the effect was going to be, then we could adjust our proofs and account for it. There are a lot of different scenarios that can affect bias in either direction. So, for instance, there's some concern that over time we may underestimate genomic predictions because the bulls that are really lousy don't ever get to have daughters. And so there's a bias toward only the best bulls get used, and so we're comparing the best to the best, and we may not recognize how truly good those bulls really are, so we could underestimate it a little bit. So I think that there will continue to be a bit of a slippage, and that we'll continue to see a little bit of loss, but by how much, we don't know for sure. Otherwise, we would correct it and take care of it that way.
Chad, how much variation exists among the Holstein populations of different countries or continents? Is it necessary to have different chips to genomically test them?
Well, first of all, there's a fair amount of difference between different continents, but not there's difference, but not extreme, because most countries have used U.S. Holstein semen. In terms of the different chips, basically these chips are designed to look at not just Holstein, not just Jersey, but beef cows, even the Indicus breeds a little bit as well. So these SNPs are spread evenly throughout the genome, and so if there are subtle differences between European Holsteins and New Zealand Holsteins and U.S. Holsteins, these chips would pick that up very well.
Chad, what impact will selection intensities, especially on young heifers, have on genetic progress regarding the bulls that are being used? Interesting question.
Yes. I think that the accuracy is the more important aspect because when we're thinking about selection in heifers, because previously based on parent average was how we selected the heifers and the cows that we would use to provide bulls to the bull stud, and we only used the best of the best based on their parent average. And so now we're genomically testing. We're still only going to use the best of the best, but we're not necessarily going to be considering a lot more heifers to be bull contract females than we were in the past. So selection intensity will change a little bit, but the bigger issue is that we identify those females much more accurately than we did in the past.
Does testing females on farms for increased selection help commercial herds increase their genetic progress?
That's a very good question, and I think, again, it's not going to change their rate of genetic progress. It can help to shrink the lag between the elite AI herds and the commercial herd a little bit more fully. I think that if you are in a point where you have lots of heifers and you don't quite know what to do with them, it's a good option. I have yet to be convinced that as part of a long-term strategy there aren't other ways to get there to the same point a little more cost effectively. For instance, if we assume that our heifers are our genetically superior heifers, which if we're making genetic progress in our herd they should be, we can keep calves out of our heifers and then sell or breed to beef our older cows and get to the same point without incurring the cost of genetic testing. With that said, I do think that every herd should at least genomically test their heifers at least once, because they're going to find out a lot about their herd. They're going to find out what their sire misidentification rate is. They're going to find out what the average genetic value of the females is in their herd compared to average. And that can help you to identify from a management standpoint, am I taking full advantage of the genetic potential in my herd? So I think it can pay to do it at least one time, but then after that, is it part of my long-term strategy, something I want to do every month, every year, from here on out? I've not been convinced that that's economical at this point.
Chad, this is Steve Larson. Let me just break in and on that same line of thought. Would that be a way where you could determine the best place to put sex semen, your more expensive semen, perhaps on the heifers, the virgin heifers that appear to be genetically superior based on genomic testing?
Well, what I would say is that you can buy semen from the best bull in the world if you're buying enough of it for probably $50 to $60, and the genomic test is going to cost you $45. So you're better off saving your money and just breeding everything to the very best.
Okay.
Interesting question. What is the value of direct genomics compared to official genomics? Does that make sense to you, Chad?
Yeah, so when we talk about direct genomic values, what we're doing is we're ignoring our pedigree information and just looking just at the DNA markers. How do they rank the animals? And so what they found is that when we include both the direct genomics and the parent pedigree information, we do a little bit better job in terms of the accuracy of our genomic prediction. However, that probably also contributes to bias, upward bias just a little bit, because a lot of times in particular the dams can be over-evaluated. So I think it's interesting to look at them and to look at the differences between the PTA and the direct genomic value. But adding that pedigree information in has consistently helped to improve our accuracy just a little bit. So I rely on that in most instances.
Well, you're actually losing ground here, Chad, if you want to know the truth. We do have a number of questions. I think we'll do about a couple more if that's agreeable, and then turn it back to Steve. Interesting one. These techniques are more developed. Will other animals like swine benefit from it? What about other livestock species other than dairy?
Well, certainly beef will benefit from it quite substantially. Poultry and swine can benefit from it, but the main benefit, remember, is that we're reducing our generation interval. And swine and poultry have a shorter generation interval to begin with. So the relative gain for them maybe isn't quite as big as it is for dairy cattle. But yes, they will benefit. The relative gain might not be quite as much as what we see in the dairy industry.
Here's our last question, and then there's a comment. Do you think that our little knowledge about DNA takes into account errors about like dairy cattle reproduction? You want me to repeat that for you, Chad?
Yes, go ahead and
do you think that our little knowledge about DNA can take in error about matters like cow reproduction? I'm not sure I understand the question. Do you?
No, I'm not. I need some follow-up on that question to know quite what they're asking.
Okay. Then the last comment is a little bit long, but we'll do it anyway. I think, to be fair, it's important to point out that the bull data comparisons is somewhat selective and biased, and genomic data calculated 3 years ago, a lot of things have happened since then. Any comments on that?
Yeah, that's why I didn't go back to 2009, because we continue to update the system. But you know, that same comment was made in 2011. It was made in 2012. It was made in 2013. It was made in 2014. But we still see the same general trend.
Okay. Well, Chad, I think we've gotten to the things we really have to hit here. Steve, we'll turn it back to you to wrap up. And,
okay.
Thanks, Chad.
Thank you, Mike and Jim Baltz down there at the University of Illinois, and again, thank you, Chad, for a great job. Obviously, there's a lot of interest. I didn't mention earlier that Chad is one of the co-authors of our artificial breeding column in the magazine, and Chad, I think you've got a lot of, based on the interest in your webinar, there's lots of things you can write about in the future. So there should plenty of ideas for future columns. And again, quickly, we thank Neogen Corporation for their support. So Chad, great to have you with us. We appreciate your participation, and we thank everyone that's been with us out there for the webinar today. Thanks so much.
Okay, thank you.
That's all from Fort Atkinson today. So.